shortchange Dr. Rudy on his presentation time, but it's a great pleasure to introduce Dr. Rudy. He's an absolute pioneer in ECGI and simulation and a lot of areas that have been historical strengths at CVRTI, areas of emphasis, and he's worked with a number of investigators here over the years. Dr. Rudy has also been influential in the field as far as training excellent investigators, including Robin Shaw, the director of CVRTI. Dr. Rudy is currently, well, he's been at Washington University at St. Louis for some number of years. Maybe we don't get into the number of years, and it was at Case Western Reserve before that, and has been influential, has been on NIH study sections and panels for many years, has had a vigorous research portfolio funded by NIH, I assume F.H.A. and other institutions as well. And as I said, has been a pioneer in the field and someone that has influenced the development of ECGI and understanding of arrhythmias to a great measure. So with that introduction, if Robin gets in, he may want to say a couple more words, but we'll turn the time over to Dr. Rudy to talk modeling and imaging cardiac electrical excitation.
Give Robin 1 minute. He's in his office. He's signing on.
Oh, okay. So Robin,
I'd like to say a few words real quick.
I figured he would when he got free would want to say something. But maybe while he's signing on, Dr. Rudy, you mind mentioning who you've worked with here at CVRTI in the past? Just some of the investigators.
My real collaboration, and I'll show one slide of Bruno, but my real collaboration was with Bruno Riccardi when he was first in Italy, in Parma, and then when he moved to Utah, we continued our collaboration. And in some of the studies, there was participation from Rob McLeod, and from Phil Ersler, and Bob Lux. Mm-hmm. And Bruce Stadman was an instrumental person in all of this, of course.
Yes.
I have a long history and a soft spot for CVRTI.
Yoram, I'm so sorry to be late.
Yes.
Great. Well, I did a brief introduction, but Robin, we figured you'd want to say a few words.
Yeah, mind.
Robin, I was introduced by Derek.
Oh my goodness! All right, I can sit back, but I have to say a few words. You know, you've never been able to keep me quiet, and why start now?
Not trying.
Thank you very much. You know, if you don't mind, I really feel like saying a few words. The reason I'm late is, as you guys know, it's the semi-annual meeting of the Nora Echoes Treadwell Foundation, and I mentioned to the board, which the meeting is still going on now, but I mentioned that I have to run away because Yoram, who's the most distinguished guest we're going to have, and also my mentor, is given the CVRTI seminar, and they're so keyed in to Yoram, and Yoram, they remember you so well. It was actually quite remarkable. And so do you mind if I just spend a minute on an introduction? Is that okay?
That's fine.
That's fine. Okay, so Derek, I'm sure, and I wasn't on, I'm sure you covered really, the obligatory highlights that Yoram, of course, did his bachelor's and his master's in physics at the Technion in Israel. If I remember correctly, it was high energy physics. Then, and this is very important, he did the first year of medical school at Case Western Reserve University in Cleveland, before then really focusing on his Ph.D., which he obtained in biomedical engineering under the great Bob Plonzi, rose to full professor in 1992, and in 2004 moved to Case Western's great loss, moved to Washington University, St. Louis, where of course Yoram is the F.A.H.A. Distinguished Professor of Engineering, Professor of Biomedical Engineering, as well as of course of Medicine, Cell Biology and Physiology, Radiology and Pediatrics. And perhaps Yoram is most known for is driving the Cardiac Bioelectricity and Arrhythmia Center there, where he is director. Yoram has been focused on two questions throughout his career, and I feel embarrassed summarizing somebody like Yoram's career in 2 sentences, but really is how to solve the forward and inverse solutions of electrocardiology. I'm sure we're going to hear about that today. And the second is to how to really compile all we know about channel biophysics and cardiac ion channels to develop a really comprehensive understanding of cellular electrophysiology and arrhythmias. And in both areas, Yoram is not just being an innovator, but frankly, he defined the fields. And he's contributed greatly to understanding of arrhythmogenesis and electrocardiology. His long publication list, and Yoram's CV is over 100 pages, packed 100 pages long. Including publications that vary including from the CVRTI, exactly, as well as papers in Science and the New England Journal of Medicine. Also Yoram, and this is very important, has trained countless successful academics. I'm not going to call myself successful, but I am going to call myself a proud trainee of Yoram. Yoram's large list of honors include induction into the United States' National Academy of Engineering. And on a personal note, and this is what I really want to emphasize, is that Yoram is a scientist through and through. If I have any rigor to my approach, I learned it from Yoram. If I have any imagination to my science, I learned it from Yoram. And you guys are going to smile at this, but if I prefer lab work over administrative work, I definitely learned it from Yoram. And so it must be emphasized that my early impressions from the CVRTI were really formed by Yoram coming back to the lab from Cleveland. This is the early 90s. After you're spending, coming back to the lab in Cleveland from Salt Lake City, where he was here working with Bruno Tacardi, Bob Lux, and others. And it is no secret, but it's worth emphasizing that his enthusiasm for the CVRTI is my primary motivator for me to come here. And I just heard from the board meeting that I must say that's a bidirectional enthusiasm. As soon as I mentioned Yoram is giving his talk, the whole board sort of erupted into fond reminiscences. And so Yoram, I hope you don't mind 2 introductions. It's an enormous pleasure that Derek and I and everybody on the call today welcome you. I hope I didn't cut into your speaking time too much.
Well, first of all, thank you, Robin, for this warm and friendly, and as you know how close we are. So thank you for that introduction. And it's true, I have a soft spot for CVRTI. I came to Salt Lake City early on and collaborated with people like Bill Berry, who was instrumental in CVRTI. And then when Bruno Tacardi moved to Salt Lake City, our collaboration that started in Italy continued with periodic visits. And I really am really thrilled that Robin has taken on the position of director of CVRTI. Knowing Robin, it will move forward with a huge forward momentum. So I can't wait to see the results of his influence. So let me start with my talk, and the title of my talk is Modeling and Imaging Cardiac Electrical Excitation. And indeed, modeling, mathematical modeling, and electrocardiographic imaging have been the two foci of research effort in my laboratory. And I would like to illustrate both through examples.
How are you guys?
Good, how are you?
How about you talk to the board?
I don't know why my, nothing is happening. Oops, okay. So these are the two areas of research in my lab. One of them is integrating cardiac excitation from the protein structure level of ion channels through the function of ion channels through the whole cell dynamic interactive environment, and then to propagation of the cardiac impulse in multicellular tissue. That's from the modeling perspective, mathematical modeling perspective. From imaging perspective, we've developed over the years non-invasive electrocardiographic imaging, or ECGI as it's known, and we use those to obtain information about the electrophysiological substrate and arrhythmias in the heart of patients in situ. And I'll show an example of this approach, the modeling approach, and the imaging approach as we continue. So first of all, mathematical modeling and computer simulations. The example will be molecular modeling of IKS, the slow delayed rectifier, potassium channels in the heart, function and its role in cardiac action potential repolarization. So why do we need to integrate? Well, many disease processes originate at the molecular scale, yet their phenotypic manifestations, in our case, cardiac arrhythmias, occur at the cellular tissue, organ, and organism levels. Integration across scales and understanding of processes that determine emergent behaviors at the higher scales, ultimately at the level of the whole heart, are essential for mechanism-based diagnosis, therapy, and what we call now precision medicine. And let me just, why IKS? Why did we choose IKS to be the example and the prototype for this approach in my laboratory? Well, IKS plays a crucial role in action potential adaptation to rate changes. You know, as we increase the heart rate and we pace faster, or when we run and our heart beats faster, we need to basically have more action potential introduced into the same interval. The only way to do it is to make the action potential duration shorter. And making it shorter is IKS, which is also subject to beta-adrenergic stimulation that is activated during exercise and emotional stress. Mutations in its molecular subunits, KCNQ1 is the alpha subunit, KCNE1 the beta subunit, cause the long QT1 and the long QT5 respectively. Long QT is an arrhythmic disease that causes sudden death. LQT1 is the most common LQT syndrome, with arrhythmias occurring under high beta-adrenergic tone. IKS is subject to beta-adrenergic regulation, and in fact, beta-adrenergic signaling molecules are incorporated into the molecular structure of IKS. IKS provides repolarization reserve when IKR, the other rapid repolarizing current, is compromised by mutation or drugs. IKR has a lot of mutations that cause the long QT type 2, and they're not only genetic, but they're also acquired by drugs. For example, some antibiotics, some antipsychotics will block IKR, and then IKS kicks in to provide repolarization reserve to restore normal reserve and prevent arrhythmias. From the practical perspective, KCNQ1 is highly homologous to KV1.2, for which there is a known crystal structure, which helped us to simulate the structure of KCNQ1. And then there is also more recently a cryo-EM structure of the KCNQ1 channel and was published recently. Both of them by the laboratory of Rod McKinnon. So in a cartoon way, in a cartoon fashion, here is the structure of IKS. IKS have 4 of those structures, which have 4 of those subunits, which is the KCNQ1 alpha subunit, that has 6 transmembrane domains. S4, okay, has arginines that are positively charged, so it's a positively charged transmembrane domain that is the voltage sensor of the channels. When you apply voltage on the channel, this responds by moving. And then it combines with the beta subunit KCNE1, which is a simple 1 domain, and the stoichiometry of the combination of these, and this is 4:2. Now, 4 of those alone, without the beta subunit, make a pore and a functioning homomeric channel. The beta subunit combines 2 of them with 4 of those to modulate and change the channel properties and channel's kinetic, which makes IKS. Without the beta subunit, IKS is the KCNQ1 subunit channel that has very different properties than IKS. So what happens when you apply voltage? Here is the cartoon of the 4 KCNQ1 combined with 2 KCNE1. This is a top view, and the channel is closed, okay? When you apply voltage, you will see S4 and somewhat S3, the voltage sensor moves up and tilts the S6, which is the pore forming domain, to allow the channel to open and move ions through to generate the IKS current. Now, S4 is going up, S6 is sort of tilted, and then the pore is open, and potassium ions can leave the cell to reduce the voltage towards the rest potential in repolarization. This is an example of a very large library of IKS channels that we constructed in my laboratory. These are millions of structures. And you can see in this example a side view. Here is the 6 domains that cross the membrane. In this purple color here, we show the KCNA1 that is introduced into this channel. The channel also contains calmodulin shown here. This is the N-terminus, and all these helices that protrude into the inside of the cell. This is the membrane. So it looks like a jellyfish, but it's really a beautiful structure, and its morphology is really anchored in its function. And here are 4 of them making a pore. So now if you expose this channel that we have on the computer to the forces, physical forces that operate in the system, like electrostatics and van der Waals and stochastic force field, the channel will gate. The voltage sensor will move and the channel will open. So we simulate. Here is one such simulation. Remember, this is stochastic. So every time we put voltage, it will happen in a different way. But you will see this will indicate an open pore. This will indicate a closed pore. And I just showed the main building blocks of the channels, not all of the atomistic details. You can see the voltage sensor here upon 50 millivolts, from minus 100 to plus 50, depolarizing pulse. This starts to go up and the channel fluctuates, but it still doesn't open, okay? That's interesting because you see the channel is depolarized, but still is not open, unlike the common understanding that depolarized channels are in the open state. They have a probability of being in the open state because it's stochastic. Now, after a while, this is even higher now, and there are more frequent and longer openings of the channel. And you can see the fluctuations of the pore. And if you wait even longer, there'll be even longer and more opening. And when you do this 1,000 times, you start to obtain what we know and is familiar to us as the macroscopic current, the ensemble current through many ion channels. Now, when this is a cartoon that shows this is the S4, the green is S4, the voltage sensor. Upon depolarization, it moves 12 angstroms up and rotates like a corkscrew. So its positive charges on arginine molecules will interact with other molecules that are charged, other residues that are charged on other subunits. For example, if you look here, you will see that R4 is close to E2, or R6 is close to E2. As it moves up, you can see now that R4 is even closer to E2, and when it moves even higher up, R2 is now close to E1, and R4 is close to E1. In other words, as it moves, there are many different electrostatic interactions that cause electrostatic potentials. So what we can do, we can plot the energy at every one of these positions, each one of these positions as a function of the height, okay, and which is we call translation, and rotation of the voltage sensor. It shows it here, rotation, translation, and the color is energy. You can see immediately that there are 3 energy minima, and an energy minimum is a stable position of the channel. So we have this metastable location, okay, with a minimum of energy that we call the deep closed state. Then there is another intermediate closed state, and here it shows the structure. And finally, it gets to here, where this is a permissive or activated state from which the channel can open. I'll show you this in an animation. Okay, so this is the activated state. This is the voltage sensor. You'll see it'll go down. This just show the structure. It'll go down, and the energy now goes to the intermediate state. And as it continues to go down, the energy will go to the remote, unavailable state of the channel, or remote closed state. So now we apply voltage on the membrane. That was at 0 voltage. Now we apply voltage on the membrane. Look what happens at minus 80 millivolts. At minus 80 millivolts, most of the minima migrated to here, to the remote closed state. And indeed, we know that the channel is closed at minus 80 millivolts, but it's really not closed. There are still minima here. So there is a probability for the channel to be closed or open, even at minus 80. But the probability and the range of the minimum here is much greater than the opportunity to be in the permissive state. So most of the time the channels are closed, although there is a probability for them to be open, even at minus 80. At plus 60, it's the opposite. The minima migrated to the permissive state, and now there is high probability for the channel to open. And so we do this, and every time all 4 channels are in the permissive state, they can open. And when we simulate it on the computer, we obtain those single channel traces that are exactly what you will see when you record from single channel IKS channels, single channel IKS preparations. So we now do this 1,000 times or 1,000,000 times, and we combine and add up all of these traces, and we obtain the macroscopic activation curve of the channel that we're familiar with. This is what we're familiar with at the activation curve of the KCNQ1 channel. Now, to abnormalities and mutations, if you have a channel and if it does what channels do on the computer, you can simulate replacing one amino acid and one residue by another to simulate mutations. So we did. And I asked my colleague, Jianmin Sui, to do the same thing experimentally in the frog oocyte, in the frog egg, and he did. So first of all, this is the wild type. This are the experimental results of Dr. Sui for the macroscopic current. These are the computer simulated results. Note that the computer simulations are more jagged than the experimental ones. This is the first time that I see this happening. Usually the modeling is smoother, but this is because we only do this 1,000 times, and there are millions of channels in Dr. Sui's preparation. If we were to repeat this 1,000,000 times, then this would be just as smooth as this. Then he looked at a mutation we call E160Q. E160 is the E1 that I show you on S2 of the channel, and it interacts with Q on the voltage sensor. And we replaced basically in a E1, okay, E1 is replaced by Q, where E1 was negatively charged, okay, and interacting with arginines on the voltage sensor. We replaced it by a neutral but polar molecule. We then replaced it again. The negative was replaced by a neutral but non-polar. And here is an extreme situation where we replaced the negative residue by a positive residue. And you see how the energy landscape changes and loses stabilizing minima, okay? And then finally, in this situation, the two residues, okay, push away each other instead of attracting each other, and the minima, the stabilization minima, completely disappear from the energy landscape. So what happens to the current? By the way, this is a clinically occurring mutation. It's the most severe clinically occurring mutation of IKS, causing the long QT type 1. So this is clinical. And what you see here, the wild type, then reduced current, both in experiment and simulation, further reduced as the severity increases. And finally, in the most severe mutation, there is no current going through the channel. Now, what else can we do beyond what experiments can do? We can integrate the channel into the cell. And we have cell models of the guinea pig was our first one, then the canine, the dog. And finally, in 2011, we published a very detailed model of the cell of the human myocardium based on cells from 144 undiseased human hearts. And you see here an illustration of the guinea pig model and many of the currents, and we can really compute all of those different repolarizing current, repolarizing current, depolarizing current, pumps, exchanger, calcium cycling, the calcium transient. What you see here from the computer is the action potential, the calcium transient that is generated during the action potential, the very sharp and high amplitude inward sodium current that causes the rising phase of the action potential. The L-type calcium current has a spike and a dome morphology. The spike is a trigger to the sarcoplasmic reticulum to release calcium. And the dome is a depolarizing current that supports the action potential plateau against the repolarizing current, outward currents of F.H.R. and IKS. So we introduce IKS or KCNQ1, both normal and mutant, into the whole cell and we allow it to interact interactively with the dynamics of all the other processes in the cell and the dynamic changes of ionic concentrations. And here are some of the results. So here is the action potential at slow rate, and here is the action potential at fast rate. The red is the wild type, and this blue here is the most severe clinical mutation. And you can see prolongation of the action potential in the mutant. And the prolongation is greater at slow rate than at fast rate. Here's a summary. You can see now the wild type, and as the cycle length is increased and the rate is decreased, the prolongation of the action potential becomes longer and longer until it's very long at slow rate. And this is really a signature of the long QT syndrome. It's more severe at slow heart rate. And then we put the cell model into multicellular tissue and compute pseudo-electrocardiograms, and you can see prolongation. This is wild type, and this is mutant. You see prolongation, or on a large scale, you can clearly see prolongation of the QT interval, giving the name long QT syndrome. So I just wanted to, I showed you one example, and now that example, there are many simplifications made on the channel structure. We assumed symmetry, we assumed concerted opening of the channel, and so on. And we also didn't take into account atomistic scale, small scale, atomistic scale movements. It was enough to simulate what we're interested in, but we decided to go further and increase the accuracy, reduce the scale to the atomistic scale, and also reduce all of these assumptions. I don't have time to show it here, but it was published. And in order to do so, just to give you a tip, this could not be done by brute force computation. And what we've done was to use artificial intelligence, machine learning, to train the system to compute the energy of structure in many, many more structures at the fine detail of the atomistic scale. And with machine learning, we were able to get very, very good results that really simulate the measured experimental results in single channel gating of IKS. So that's the first part of my talk, and that's about some examples of how we use modeling to understand mechanisms. But at the whole heart level, we shy away from developing models of the whole heart because the whole heart is complex, and existing models of the whole heart really use severe assumptions. For example, differences in conduction system between individuals are not incorporated. Differences in expression systems and its heterogeneous spread throughout the myocardium, those are not taken into account. So we decided to go in the way of basically recording non-invasively the electrical activity of the heart in patients. And that's where ECGI came in, and I've been involved in developing ECGI for many, many years for mapping the arrhythmic substrate in patients. And what is ECGI? I think many of you know, because there is work going on also at CVRTI, I believe, with ECGI. But anyway, what we do is we record 250 electrodes simultaneously from the torso of the patient, front and back. And if you pass one line through all of these electrocardiograms, you'll obtain the potential distribution at that instant of time on the body surface. We do this every half a millisecond throughout the cardiac cycle, or many cardiac cycles as needed. And that will give us the entire electrical information on the body surface non-invasively. We also push the patient through CT or MRI scan to obtain the electrode positions on the torso and the anatomy of the heart to generate a three-dimensional heart torso geometry, which is patient-specific. And then here went many years of development of methodology and algorithms and validation of what we've done. And the consequence of all of this was the computation of potentials on the surface of the heart. It's a epicardial surface, okay? And then from this, you could compute as a function of time at any point on the heart surface an electrogram. From the electrogram, you could obtain the activation sequence, which is the isochrone map, and the repolarization pattern, okay? And also ARI, which is the distance from activation to repolarization, which is shown to be a good surrogate for action potential duration. And here, I really want to dedicate this talk to Bruno Teccardi. I think many of you knew Bruno. Bruno was a dear friend, and we shared not only passion for science and the heart, but also passion for music. And Bruno passed away in August 2015 in Italy. Anyway, we started our collaboration for validation of what we've done at Case Western Reserve University then, with an existing tank torso that was in the University of Parma in Italy. And some of my students went to Italy to participate in these experiments. And then when Bruno moved to Salt Lake City, this is a picture of the tank torso he built with Bruce Stedman in Salt Lake City. And here are two of my Ph.D. students analyzing data. It was a very fruitful collaboration that allowed us to validate in a direct fashion the accuracy of our algorithms. And then we did evaluated the same algorithms over at Case Western during open heart surgery. This time we put the electrode in a vest, and we had two electrode carrying arrangements here, two patches. One was anterior, one posterior on the heart in the open heart surgery in a patient. We recorded directly from the heart, we recorded from the body surface with this vest, and we compared the reconstruction with the directly measured electrograms. And here is an example of the anterior patch in one subject, and you can see these are the non-invasive reconstruction, and these are the invasive measurements at the same location. It's not exact, but it's very, very close. And that convinced us that we could start after the validation in the tech torso and in patients, we started to use it to study the mechanisms of arrhythmias and the substrate in humans. So the examples I wanted to give are post-myocardial infarction electrophysiological substrate for ventricular tachycardia. And post-MI substrate is an anatomical scar. And the electrophysiological signature of a scar are low level, very low level, fractionated multiphasic electrogram. Why? Think about the scar as an inhomogeneous substrate of islands that are surviving myocardium, separated by connective tissue septa. So one island is activated, it's small in volume, it generates a small deflection, then there is a long delay to the next island and so on. This is known as discontinuous conduction. And that slow discontinuous conduction is sufficient to separate the electrogram into multiple deflections, but there are low potentials. In contrast, if you are away from the scar, you have a single deflection of a robust, high level, high voltage electrogram. So we grouped together all of the fractionated low-level electrogram. You can see this one, for example, on an expanded scale here, and you can easily see 1, 2, 3 deflections. When we combine them all, we obtain this red region here, which we call the electrical scar associated with the anatomical scar. The green is graded. There is a little bit higher potentials, less fractionation. That's the border zone of the infarct. And the border zone is the source of many arrhythmias, is known from studies by Andy Wheat and Penny Boyden at Columbia University, New York. How do we validate that what we've done, that the substrate we constructed is correct, we have to, it has to be consistent with the anatomical scar. The electrophysiological scar is generated by the electrophysiological scar. So they have to be, the electroanatomical scar and the anatomical scar have to be consistent. So we used delayed contrast MRI with gadolinium to map the anatomy of the scar. And this is the epicardial aspect of the MRI reconstructed scar. We then co-registered it with ECGI, and now you can compare the two, and you see how beautifully, okay, the electrical substrate follows the anatomical scar. And I can show you more examples of this with different anatomy. Here is an apical scar and its associated electrical scar. Here is a more complex anatomy of an anatomical scar. And the electrical substrate really follows and superimposes on the anatomy. So it's all very consistent. One more property of scar is late potentials. During sinus rhythm, there is slow, discontinuous, lingering conduction after the main QRS in the scar. And you can see here in the frames what is known as late potentials, and all of them, all the late potentials that we saw came from the scar. And here I wanted to show you an example of reentrant arrhythmia. Arrhythmias in broad strokes can be divided into reentrant arrhythmia due to the formation of reentry circuits or rotors, and focal due to the abnormal firing of a group of cells that become a focus or a pacemaker that drives the heart. These are the two, in broad strokes, mechanisms of arrhythmias. So I wanted to show you an example of ECGI reconstructed reentry. This is in a patient with inferior left ventricular scar. The scar was imaged with SPECT, shown here in blue, and it's shown here on the ECGI image in red. And the reentry circuit is here, right here on the epicardium. You can see the beautiful rotor that goes around and around using the scar as an anchor. The patient was ablated here with catheter ablation, and this arrhythmia was terminated successfully. And I'll show you this in animation. Here is one VT, one ventricular tachycardia rotational beat. Then there'll be another one. There'll be an exit site from the scar here and another rotation. Now it will be spontaneously interrupted by a non-VT beat, and then another VT beat, another rotation. Okay, the other type of arrhythmia that I was talking about is focal. And here I wanted to show something that is really textbook physiology, the concept of extension of the line of block. So here is a patient, okay? Here's a patient. This patient has non-ischemic cardiomyopathy, so it's diffused fibrosis throughout the heart. And he has a line of block right here. You can see the black line of block. This is during basic pacing from here with what we call S1 pacing, that is a steady pacing at a certain rate. But then we give a premature beat, S2. And the beat is premature. What does it mean that it's premature? It means that sodium channels that were inactivated during this beat did not have enough time to reactivate, to recover from inactivation. So there is less sodium channel availability and therefore less excitability. So now the activation before went around the line of block, and you can see here very crowded isochrones, which indicate very slow conduction. So here is an Achilles heel of the substrate. Very slow conduction, meaning it's compromised. And when we now reduce excitability by premature beat, it will be compromised even more. And what was very slow conduction becomes a conduction block. You see, this is called extension of the line of block and was really demonstrated many times in animal experiments. But this is the first time that I've seen it demonstrated in patients. Okay, so now we pace from here with another premature beat. Now activation goes all around this longer line of block and ends down here in this region. And when we let go and we don't pace anymore, then activity starts all of a sudden from where the previous beat ended, the paced beat ended. This suggests triggered mechanism as the mechanism of this focal arrhythmia. And that becomes the source of the VT, and it's steady, it's persistent, and monomorphic. Every beat is the same. I'll show you this also in a movie. So here is one paced beat, starting from here where the pacing electrode is. Another one coming. Now the S2 premature beat. We'll start from the same location. We didn't move the electrode, but it'll terminate lower. You'll see now it'll go through higher through the heart and will terminate down here. Another one. And then we let go. After this one, we let go. And these are the two VT beats that are shown. And you'll see they start from where the last one, that's where it ended. Look now, started from there. Okay, it's another one. Start from where the pace beat ended. Okay. The previous examples focused on activation and abnormal conduction, but repolarization is also very important. As I showed you in the first part of my talk, long QT syndrome causes arrhythmias because of abnormal repolarization. So I wanted to show an example of ECGI of abnormal repolarization. And indeed, I'm closing the loop in some way because I saw molecular simulation of long QT. Now I'll show you a true long QT substrate in a patient. This is a normal heart, and the activation recovery interval, which is measure of the local action potential duration, you see is all blue and homogeneous and around 240. This is LQT1, which is the IKS, the related long QT that I showed before. And you see here that in this whole region, the action potential duration is greater prolonged. It goes from 240 to 400 or 420. Very, very prolonged. And it borders with, or it neighbors with, a region of much smaller action potential duration. So it's not a homogeneous prolongation. And as a result, it creates a region of very steep repolarization gradients, okay, known as steep dispersion. Now, an action potential triggered in this region cannot enter this region because this region has long action potentials and will be, at that time, will be refractory. So it cannot enter, this will be a block, and it'll have to go around it and will return to form a reentry circuit. That's the danger of this arrhythmia. The substrate is there waiting for a trigger. So in numbers, here, the dispersion maximum is 7 milliseconds per centimeter, while here it's 104 milliseconds per centimeter. Huge difference. It's 2 orders of magnitude. And interestingly, the steepness was greater in symptomatic patients, was 130, versus only 98 in asymptomatic patients. That suggests that ECGI mapping of the substrate could be an index of vulnerability to life-threatening arrhythmia in the long QT syndrome patients. So just to close, I'll show you where we are now in ECGI. First of all, non-invasive ablation. This was published, oops, I'm sorry. This was published some time ago in the New England Journal of Medicine. And since then we had several other papers. But anyway, what we do, once we know from ECGI where the focus of the arrhythmia is. And we also do MRI scan to get the scar substrate associated with this. Then we irradiate it with ionizing radiation, X-ray radiation. That's the usual treatment, planned treatment that they do for tumors in cancer patients, in radiation oncology. And this could be very precise these days with the technology of today. And I just show you the first results from the New England paper. There are 5 patients. These are the burden of VT episodes they had, and look what happened after up to a year. Really, they had no arrhythmias. I mean, the procedure is very simple. We do ECGI, and then a day later, the radiation takes about 10 minutes, and the patient is awake. It's not like ablation with a catheter, right? It's non-invasive. And the results are dramatic, really dramatic and very promising. So now there is effort in our institution to study the mechanism, what happens. And some initial results just published in Nature Communication show that there is an overexpression of connexin 43 or gap junctions post-radiation, and also overexpression of sodium channels. And optical mapping shows an increase in the velocity of conduction. So that could be part of the reason why it is so successful. The other thing that we've done was combine imaging modality. Here it shows ECGI, this is potentials, with the asterisk showing the breakthrough site on the right ventricle. These are isochrones, early activation, also at the breakthrough site. These are repolarization. By the way, activation recovery intervals or action potential are longer in females than in male, 263 versus 232. But what we did that is novel was, these are all normal volunteers, 20 of them. And what we did, we also recorded the contraction of the left ventricle using tagged MRI. And we combined the two together, so you can see excitation and contraction in the same heart in situ. And one thing to pay attention to is that the time scale. The excitation, electrical excitation, is almost instantaneous throughout the heart, very fast. But to me, it was surprising to see how much slower contraction is, very different time scale. So here is this movie, and I'll end with this movie. This is RV lateral view, which is probably not the best view to see it. You'll see later in different, see, that's the excitation, so fast. And now comes contraction. If you see how, this is the actual movement of the left ventricular wall in this particular subject. Now it's feeling, relaxation and feeling. You'll see how much slower the mechanical process is compared to the electrical process. LV lateral view probably will show it better. And then, so here's LV lateral view, excitation, very quick, gone. Now contraction, much slower. See how the ventricle becomes smaller, squeezing the blood, and then refilling. Okay, let me stop here. It's too long of a list to read everybody, but these are the people who contributed to the modeling and experimental work in the cells. And these are the people who contributed to our non-invasive mapping. And I thank you for your attention. Thank you very much.
Thank you, Dr. Rudy. Robin, do you want to handle questions, or do you want me to?
Go ahead, Derek. Thank you.
Okay. Well, Dr. Rudy, wonderful presentation. Thank you so much. Hard to summarize decades of excellent work in a 45-minute talk. We got a few minutes just for a few questions, but can I start by asking one? you mentioned that you've got some of the best cellular models that you've developed with your lab, and now in humans, you've done dogs, guinea pigs, the whole range. But you said there are problems with implementing that as a whole heart simulation. What is it that we're missing? Computing power is up. We can make very complex models. Are we missing anatomical information? Are we missing what is it that holds that back?
So here is I'm talking about simulating the whole heart for the purpose of understanding mechanisms of arrhythmias. Okay. You have to remember that arrhythmias really are generated by single cells and the tissue. Okay. And the existing models of the whole heart, some of them claim that, you know, they do things on the cellular scale. Well, they don't. They have much larger elements that they use for the whole heart. And there is a lot of very important, crucial information for the mechanism of arrhythmias in a particular patient that is not incorporated, and we don't know that information in a particular patient. For example, the heterogeneity of expression of IKR. We know that IKR is heterogeneously distributed from the work of Gene Nerbon, let's say, throughout the heart with really major differences. And that will, of course, contribute to the normal and abnormal properties of the excitatory substrate. Similarly, it has been shown that in, let's say, catecholaminergic polymorphic ventricular tachycardia, CPVT, the Purkinje muscle junction is essential in formation of the arrhythmia. And, you know, just like you and I don't have exactly the same fingers, we don't have the same conduction system. And that makes a big difference where those, to map the conduction, we don't have maps of conduction system specific to a patient. I can go on and on, the distribution of gap junctions, all of those things. So the heart models that they say that they're patient specific, they're patient specific as far as the gross anatomy and maybe also the fiber orientation in the myocardium. That's what they call patient specific. But to be patient specific takes much more than those gross properties. And the microscopic properties are crucial in the generation of arrhythmias. So I don't know, I may be wrong, but my philosophy was, if, you know, nothing is better than trying to map the actual heart of a particular patient and not try to simulate it. But, you know, this is my personal opinion.
Great, thank you. Do we have other questions from members of the audience here? Jake made the comment in the chat that torso tank is downstairs in the basement next to their working area, so it's still sitting there. So, yeah.
I know the last time I was there, they made sure I'd go down to the basement and see it.
Yeah. Okay. I can't see most people, so if anybody has their hand raised, I guess I don't see it. Well, one other question that I have, if other people don't have questions, is, you know, you talk about ECGI and then using it with this irradiation method, and they can be focused with the irradiation. How focused do you need to be, or can you kind of carpet bomb a whole area? And is the precision not required like you need with RF ablation, where you have to get exactly where you're going to be? With the irradiation, can you hit a bigger area and be effective with the therapy?
So right now they irradiate a larger area, mostly because the heart moves and the cardio moves. So they cover I don't know exactly the volume, but it goes across the wall and cover it as an area of the scar, but not the entire scar, only where the arrhythmia is originating. Otherwise, you know, they're very complex scars, and they're scars with multiple locations, and you cannot irradiate them all if they're not arrhythmogenic. That's the benefit and beauty of ECGI, determining which part of the scar is where the arrhythmia is coming from. So, but I was told that there are now available already, or it's coming soon, the possibility of taking, compensating for the movement of the heart. Then it'll be better to focus. We don't know. There are many practical questions and also many mechanistic questions. The only mechanism that we know what we know is that the arrhythmia ceases to exist, it terminates much before there are any structural changes to the tissue because of irradiation. So it's not the cause of fibrosis or apoptosis that is causing it. There are other physiological changes due to the radiation. It's very interesting. And the one that we found here and also was found earlier by a Japanese group is proliferation of connexin 43. Robin will find this interesting. And also proliferation of sodium channels. And the sodium channels and gap junctions are in the same location in the heart at the intercalated disc. So it's intriguing and interesting, and I think it's just the beginning of a story that will not only make it possible to do non-invasive ablations as routine, but also will teach us a lot about the heart electrophysiology.
Great. Well, I apologize. I think I took the time with the questions, but I think we need to move. You've got another group that's going to meet with you here in a moment. But thank you very much, Yoram, for an excellent presentation and for your great work. Sure appreciate it.
Absolutely. Thank you. Thank you for having me.
Thank you.